My name is Tim Mitchison. I'm from Harvard Medical School. I've been working with fluorescent probes since before we had fluorescent proteins, or at least before they were widely used, and have done quite a bit of chemistry and that kind of thing on them myself. And I really love these molecules. One of the things I'd like to give you guys is some appreciation for these beautiful molecules and their properties, and particularly how their chemistry gives rise to their properties. So, but for those of you who aren't chemists, don't worry, I'm going to try and make it as easy as possible. Okay, so the outline of this presentation, we're going to be talking about probe chemistry, and then we'll talk about protein labeling, and then finally non-covalent targeting of probes. Okay, and in the chemistry section here, I want to talk about the structure of probes. I want to spend quite a bit of time on that. People buy a probe from a catalog and they attach it to a protein, they never really think about what that molecule is. I'd like to encourage you and give you some tips for really looking at and appreciating these molecules. Talk about how the wavelengths is determined, just briefly mention quantum dots, and then issues like photobleaching and photodamage. Okay, so we'll start here. This is really, I want to sort of how to look at probe structure. I think a nice place to start is with this beautiful molecule here, Fluorescein, arguably the first synthetic probe synthesized in 1871 as part of the German dye industry that was going on at that time. So people were building dye molecules for commercial applications, and this is sort of like a dye molecule. It's highly colored. And fluorescein is still in use today. It's, for example, it's a very cheap fluor and non-toxic. You can use it to trace flow in underground rivers. And it's a molecule I've made myself many of times. It's actually one of the most fun things I've done in science is you take two white molecules and you cook them up in a strong acid overnight. You come in the next day and you've got this brilliantly colored solution. And if you put a drop of it in water, the whole thing goes green fluorescent. So it's a really very fun kind of molecule. Now, the fluorescent properties of fluorescein are pH dependent. This form over here is with the O-, which is the high pH form, is the fluorescent one. You can see here the excitation and emission spectra. And so I'm really only going to be talking about this one, although this ability to switch between fluorescent and non-fluorescent has many applications. This is, I hope, review. You should have heard about Jablonski diagrams in Nico's talk when he was talking about the principles of fluorescence. Here, I really only want to focus on the HOMO, the highest unoccupied molecular order, and the LUMO, the lowest unoccupied. And of course, the energy difference between these is what determines the color or the wavelength of the fluor. The smaller the energy difference, the redder the fluor. And I want to relate that to the structures. So the part of the fluorescein molecule that really matters for its fluorescence, this lower ring system doesn't matter. That just, you can use that for attaching to proteins. It's this other part here that you can see extending from this O- up to this carbonyl here. And if you look, there's alternating single and double bonds here, which means it's a conjugated structure. In fact, electrons can be in an orbital that extends across this whole structure here. We call this an extended orbital. And it turns out that the larger this extended orbital, typically, the lower the difference between the HOMO and the LUMO, and the redder the fluor. You also notice almost all fluorochromes have an electron donor at this end, O-, an electron acceptor at this end, the double bond O. So you've got a kind of a push-pull system. But this is a symmetric push-pull system. The two are essentially the same here. And this symmetric conjugated orbital, it turns out, reduces quenching by other dipoles like water. You can also have fluorescent molecules with a permanent donor at one end, like an amine, and a permanent acceptor at the other end, like a ketone. Those can be very fluorescent in organic solvent, but they don't tend to fluoresce as well in water. And it turns out this symmetry here is also responsible for the fairly small Stokes shift, the small difference between the excitation and the absorption. And the one other notable point about this structure, if you look at this extended orbital here, there are no rotatable bonds. The whole thing is rigidly locked into these three rings. And it turns out if the molecule can rotate, it tends to be a way it can lose energy and become less fluorescent. Now, first, let me talk a little bit more about this size issue. Compare two molecules here that I hope you can see, whether you're a chemist or not, are really fairly similar. Fluorescein, and here this is a coumarin derivative. and basically you've chopped out one of the rings and getting from here to here. And if you look at the fluorescence, this is one's 488 exciting, 510 emission. This is considerably shorter, and that's just because the conjugated orbital is smaller. You can actually add another ring into fluorescein, you get something called a naphthyl fluorescein, and that will shift it out into the red. So size is one factor. Another factor, the actual atoms matter though. Here's two more similar molecules, fluorescein we're already talking about. This is another similar molecule, I call it razorufin. I'm not sure if razorufin is more correct, but you can see in place of the carbon here in fluorescein, there's a nitrogen, and it turns out that shifts the wavelength considerably into the red. And I don't understand the kind of quantum mechanics that goes on, but that's often the case with heteroatoms. So you've seen here two ways you can kind of tune the wavelength by changing the size and the atoms. And then here's the point about rigidity, comparing fluorescein to another closely related molecule, phenolphthalein. The only difference here is this bridgehead oxygen is missing, and it means you can rotate around this bond here, so these rings can twist. As a result, phenolphthalein is highly colored. In its high pH form, it's purple. It's used as an indicator dye, but it's not at all fluorescent. Basically, the LUMO can lose energy very rapidly by a rotational process. So rigidity matters as well as size. And basically, by tuning those kinds of properties, the size of the system, the heteroatoms, and keeping everything rigid, it's been possible for chemists to build a series of dyes that cover the whole visible spectrum, all the way out here from the ultraviolet into the point where water starts to absorb. So this is the useful spectrum we have in microscopy. This particular set here is the Alexa dyes made by molecular probes. I'll show some structures of those, but there are comparable dyes made by other people. One point I want to make, you often see diagrams like this. These are the absorption spectra, and they've all been normalized, so they have the same height. In fact, the absorption coefficient, the efficiency of absorbing light, scales strongly with the size of the orbital. So these ones absorb light much less efficiently. They've got absorption coefficients around 20,000, whereas up here with these long wavelength ones, you can get as high as 200,000. So in fact, the longer wavelength fluors are much better at capturing photons and much brighter on a per molecule basis. Here's an example of one of these long wavelength ones. This is a particular favorite in my lab, Alexa 647. This is a cyanine type dye. You can see here this really extended orbital, and again, as symmetrical. This nitrogen is donating, this one's accepting, but they're symmetrical. And so this is the fluorescent part of the molecule, and I'll come back to this molecule later and talk about some of the other features of it, which are important for practical use, like labeling proteins. I just want to mention very briefly a completely different kind of fluor here, Q dots, that have become very popular for certain kinds of experiments. They look nothing like the molecular fluor. They almost look like a little tiny virus or something. Inside a Q dot, there's a semiconductor nanocrystal. It's actually a very special kind of crystal with two different layers of different semiconductors. And these core shell nanocrystals can be highly fluorescent if the band gaps of the semiconductors are tuned right. So these can be incredibly fluorescent molecules. They are nanometer size, these Q dots, so they're more like a small protein in size, and then they have to be coated with a biocompatible, this particular one has been coated with a lipid so that they don't interact non-specifically with biomolecules. And it turns out with quantum dots, you can choose tune the wavelength simply by the size of the semiconductor crystal. So you can here go from blue to deep red simply by increasing the size. And they have very, very tight emission spectra, so you can, in principle, pack more, deconvolve more of them in a microscope. Amazingly, with these molecules, they don't photobleach at all, so they don't have the same kind of photochemistry I'll talk about. So they're extremely bright and they don't photobleach, which is incredibly useful for some applications, for example, tracking single molecules on the cell surface or certain biophysical applications. The only downside of these Q dots, they are rather large, so you have to figure that into your calculations, and also modifying them for labeling proteins is challenging, although people have worked out methods of doing that. So not quantum dots, but the organic fluors do tend to photobleach, as I'm sure many of you are already aware. The chemistry here is actually not terribly well understood, but the basic idea is that the probe in its excited state can interact with molecular oxygen. Molecular oxygen is a stable radical, and it can pick up energy from the excited state, and the oxygen can be converted to highly reactive species, singlet oxygen or hydroxyl radicals. This is OH dot, not OH minus. These are highly oxidizing species. They can come back and react with the fluor and destroy it. Importantly, they can also destroy nearby proteins and lipids. So photobleaching and photodamage tend to go together. Usually we think of them as a bad thing. They get in the way of our doing an experiment or cause artifacts. But there are some experiments where people have put them to use, notably in this so-called Cali technique, chromophore assisted laser inactivation invented by Dan Jay a few years back, where the basic idea is if you have a protein with a probe on it, if you excite that in the presence of oxygen, you actually destroy the protein, or in some cases you can destroy a whole cell, and it can be used to locally ablate. So this is putting the damage to good work. And there's a version of red fluorescent protein, so-called killer red, which will do the same thing. So I'll sort of end this part of the chemistry section by saying there's been a lot of development on probes, but I think there's still a lot to do. And this is sort of attention chemists here, because I think there's still a great deal to be done by chemists in this space. Perhaps particularly I'll point out here with special properties. Probes that will switch on and off with different light, or switch on when enzymes attack them, and that kind of thing. So I think a really cool area for people with an interest in synthetic chemistry to think about. Okay, so that's the probe itself and its fluorescent properties. Now I want to talk, of course, about attaching to the proteins, because in most applications people are attaching these probes to a biomolecule, usually a protein, sometimes a nucleic acid. I'm only going to talk about proteins, and there's really two amino acid side chains we have to worry about or think about for proteins, and I'm going to use the single letter abbreviation. So C is cysteine, SH, and K is lysine. And cysteine labeling is ideal for site-specific labeling if you can engineer it, or nature gives you one reactive C per protein. Lysine is a much more common amino acid. Most proteins have a lot of lysines, and this is ideal for general purpose labeling. You can label a few lysines on most proteins without losing activity. So an important point for both these protein nucleophilic amino acids is they're only reactive in their deprotonated form, i.e. at high pH. Cysteine is reactive as a thiolate ion above pH 8. Lysine is reactive as the free amine. Although you only completely deprotonate lysine above pH 10, you usually do lysine labelings around pH 8 or something like that. There's enough unprotonated lysine to react. These two nucleophiles have very different properties, so we can distinguish them with different probe chemistry. The thiolate ion, cysteine here, likes to react with these kinds of electrophiles. So classically people would use iodoacetamide or N-ethylmaleimide. This is a so-called Michael acceptor to label protein. And this same chemistry, which goes back many years in protein chemistry, was adapted to fluors. So here are two typical thiol reactive probes. This one up here, pyrene iodoacetamide, has an iodoacetate that the thiolate attacks. This one has a Michael acceptor. So that's cysteine. The unprotonated form of lysine is what people call a harder electrophile. It prefers to react with sp2 carbon electrophiles, particularly reactive carbonyl derivatives, where X here is a good leaving group. And you want a leaving group that leaves well, but not too well, like chloride, because that would react rapidly with water. And a lot of probes now use this N-hydroxysuccinimide ester, which has a reasonable balance between being stable enough, it's not immediately hydrolyzed in alkaline solution, but reactive enough that it rapidly reacts with lysine residues. So this has come back to the probe I mentioned before, Lexus 647, to highlight a different feature of this. I talked about the orbital before, but this is the protein labeling part, and you should be able to recognize that this is an N-hydroxysuccinimide ester. The lysine attacks at this carbonyl and kicks out this NHS portion. So this is a lysine reactive probe. And the one other thing I want to point out, a lot of modern probes have these sulfonic acid groups. There are 4 on this one. And these are not reactive, they're just present as a sodium or potassium salt, and they're there to make the probe water soluble and prevent it sticking hydrophobically to protein. This one has 4, it's more common to have 2. But in my lab's experience, almost always you want to probe with some of these sulfonic acids on them. They're much better behaved, they're just less sticky in general. Okay, so that's labeling amino acids. With cysteine, you can often target to one particular amino acid. But if you really want to label a protein in one place accurately and know exactly where that place is, there are some other modern technologies for doing that that are very cool, that I'm calling here SNAP, Palo, and flash tags. And I think there are other methods being invented all the time. The basic idea here is that you genetically engineer your protein. You put some tag on it, will then act as a kind of synthetic acceptor for the fluor for covalent reaction. I'll only show the chemistry, I'll show the chemistry in detail here for the SNAP tag. Here we're using a substituted benzyl guanosine. And this part here of the fusion protein is a modified DNA repair enzyme that evolved in nature to remove benzyl groups that happen to get on DNA. It's been mutated now, so it reacts but can't unreact again. And so these authors developed this as a really efficient labeling method called the SNAP tag. The halo tag is a comparable method that uses an alkyl halide derivative. So these are ways where you genetically engineer your protein, then you know exactly where the fluor will go. They work well in cells. They also work well with pure proteins. And finally, a really cool piece of chemistry from Roger Chen, who's kind of the hero of fluorescence in biomolecules. This flash molecule, this is fluorescein with 2 arsenic groups on it here. And arsenic likes to bind to thiols, and Adams and Chen developed this flash probe that will, with very high selectivity, bind covalently to 4 cysteines engineered into an alpha helix. And here's the lead reference on that for people who are interested. It's a very elegant targeting chemistry. And I'll end here by talking, not all probes are targeted covalently. It's often very useful to have probes that target with high affinity non-covalently. They can be easy to use. I'll show just one example here. Here's a cell stained. It's probably a fairly familiar image to cell biologists, with DAPI to stain the nucleus blue and rhodamine phalloidin to stain the actin filaments red. I'll just show you how the rhodamine phalloidin works here. So, as the name implies, it's a bifunctional molecule. This is the rhodamine part, a fluor which excites in the green here at 546. And then this, a slightly more complicated piece of molecule here, this bicyclic molecule, is a toxin, phalloidin, from Amanita phalloides, the poisonous mushroom, the death cap mushroom. And this part binds very tightly to actin. So this bifunctional molecule will find actin filaments and bind very tightly to them. And you can make fluorescent derivatives of other drugs as well. It's a very useful way for localizing receptors. I'll end here on organelle probes, which is another non-covalent targeting method. Here's a pretty image of a cell, again stained with DAPI, but the mitochondria, have been labeled red here, the sort of wormy-looking structures, and the lysosomes have been labeled green. And these probes are just making use of the physical biochemistry. Both mitochondria and lysosomes pump protons, but in the case of mitochondria, it ends up as not as a pH gradient so much as a membrane potential. In lysosomes, there's actually a pH gradient, and you can use this proton pumping to specifically target these organelles. Here's a mitotracker dye that was used in that image. This is very like a rhodamine dye, but it's lost the carboxylic acid here. So this has a delocalized positive charge, this molecule. It's lipophilic, has a delocalized positive charge, so it follows the membrane potential and enters mitochondria. And then this part here, this chloro group, makes it reactive with thiols, so it will actually get trapped inside the mitochondria. And finally here, I want to end here. Sorry, I'm blocking one of these molecules, but I'll step off the screen in a second. Oops, there you go. Here's 3 molecules for you to think about. And I'm not going to tell you the answers here, but if you're interested, try to think about what kind of wavelength would you predict these molecules fluoresce at? Will they react with proteins? What other chemical properties can you note about them to predict their behavior? the one you have to stand off here to look at, this one's going to be a bit hard and is kind of a trick question, but if you compare it to the low pH form of fluorescein, you may be able to figure it out. And I don't know, I hope, especially for those of you who might be a bit afraid of chemistry, I hope this will convince you that it's actually fun to look at molecules, and if you make a habit of it, they'll talk back to you. So thank you.